All right, welcome back, everybody, to Bio 153 Online. And I'm streaming live from my house as we go through Chapter 28 together. So Chapter 28, it's a pretty lengthy chapter. And so the first part here we're going to go over is the male reproductive system. So understanding the anatomy and the physiology of the system is what our focus is going to be today. So without further ado, here we go. All right, so the first important thing here, we're looking at our notes, is ultimately looking at the internal genitalia and eventually external genitalia of the male. Of course, what's very important to know is the ultimate gonads of the male. So looking at the testes, so this white circular structure here, and within the testes is where sperm is going to be produced. And we'll talk about the precise location of the sperm very soon. So when the male experiences ejaculation during sex, there's a particular route that the sperm is going to travel. And so we're going to take a look at that here. So, sperm, the route, will leave a term, what's known as the epididymis. The epididymis. And this is where sperm is stored. So the sperm is produced within the testes, and then storage is the epididymis. So here's the testes here, sperm produced here, and then makes its way into this structure. This is the epididymis right here. This is where the sperm is stored. When ejaculation occurs with the male during sex, this structure, the epididymis, is going to undergo very strong rhythmic contractions. And based off the rhythmic contractions of the male's epididymis, the sperm will leave the epididymis and will make its way into this long tube, slender structure, and this is known as the vas deferens. Okay, this is the vas deferens here. So, vas deferens number 2. After that, the sperm will then travel to a larger bulb area, which is known as the ampulla. So, the ampulla of the vas deferens. Okay, so number 3 is the ampulla. Ampulla, right here. From here, the sperm will then make its way into what's known as the ejaculatory duct. And it's there where a lot of contents will be spilled out from the prostate gland and items spilled out from the bulbourethra gland and also from the seminal vesicle, forming semen, which contains nutrients, things like that for the sperm, to keep the sperm healthy. So here we are at number 4, ejaculatory duct. From there, the sperm now found within the semen will make its way into the urethra. The urethra is made up of 3 major regions. So the prostatic urethra found in the prostate gland, membranous urethra, that's a very small piece, and then finally the spongy urethra. So we can see that here on the diagram if we trace this. So here we are, we've made it into the prostatic urethra. Next, this small area, membranous urethra, that is pretty much between the prostate gland and the start of the penis. And then finally, making our way into the membranous urethra, spongy urethra here, spongy. And this goes through the penis all the way to the end of the penis. And then finally, the semen with the sperm goes out of the opening of the penis, which is the external urethral orifice. An orifice refers to opening. Okay? So very important to know the route of the sperm. So if I were to erase this and go through this one more time with you, because this is really important for you to know. We'll go through this here. We'll erase this. Here we go. Erasing all that for you. There we go. And we go through this again. So sperm produced right here in the testes will make its way to this structure here known as the epididymis. This is where sperm is stored, the storage location. Then when ejaculation occurs, when the male is having sex, the epididymis goes through strong contractions, expelling the sperm into this long tube-like structure, which is known as the vas deferens. The vas deferens. So sperm travels through that structure, makes its way into this bulb area, number 3, the ampulla of the vas deferens, right here. The sperm then empties into the ejaculatory duct right here. And this is ultimately where the sperm is then made up of the semen, which again has nutrients for the sperm, other fluidity. And then the sperm will make its way into the urethra. The first section of the urethra is called the prostatic urethra in the middle of the prostate gland, before eventually making its way into the membranous urethra, this area right here, before eventually going into the spongy urethra, which extends and goes through the middle of the penis, all the way down like this, and then eventually out through the external urethra orifice, the opening at the end of the male's penis. So now we're going to take a look at the scrotum. The scrotum. So here's the scrotum right down here, this area here. And the scrotum is made up of several different types of muscles. One is known as the dartos muscle, which are the wrinkles of the scrotal skin, superficial. So kind of see that down here, the dartos muscles. And then there's also what's known as the cremaster muscles. And the cremaster muscles will elevate the testes. And they're bands of skeletal muscle, so the cremaster muscle. So right over here, the cremaster muscles. Now, what's interesting about this area, too, of the scrotum, which of course contains your 2 testes, most men are born with 2 testes, some aren't, but most are, is that there's actually a temperature difference here in the scrotum versus the rest of the male's body. Actually, a difference of 3 Celsius. And the reason for that, maybe you know the reason already, but maybe not, why the scrotum is cooler versus the core of the body. It has to do with the sperm being viable, okay? So it's all about the sperm. You want the sperm to be viable. And that's important, if you're trying to have a child. Sperm must be viable, okay? So it's all about the overall health of the sperm. So the sperm needs to be alive. It needs to be viable, okay? It needs to be viable. So healthy, viable. So that's key. So that's why sometimes when a couple's trying to have a child, they're maybe having complications. It might very well be because of the man. Studies have shown that a male that goes into a hot tub, for an example, and goes into a hot tub, maybe the hot tub's cranked at 104 Celsius or higher, that male, if he's going in there every night for 15 minutes or whatever, it can actually kill the sperm or make it not very healthy. So that's a proven thing. Even the type of underwear the male wears can play a role in the sperm being viable. If the male's wearing very tight underwear, actually that can play a role in viability of the sperm if the male's wearing more of briefs versus boxers. So there's studies of that too. And they say that it's important also for the male to even drink coffee can be important. Even a little bit of the caffeine can actually play a role in the sperm being viable, as that can increase the chances of pregnancy with drinking coffee. And that sperm, even the female system, actually has olfactory receptors to sperm and actually have a sense of smell, and they can sense their way through the female reproductive tract to eventually find the egg. So very interesting in that regard. What you also find in this area of the scrotum, you have what's known as the spermatic cord. So this contains nerve fibers, blood vessels to this area, even lymphatic vessels too. Taking a look more closely now at the testes, a lot of small, very intricate structures here. So first we have here the seminiferous tubulo. And the seminiferous tubulo or seminiferous tubulos are going to be all of these dark yellow kind of curved-like structures, wavy. They're extremely important because this is the precise location of where sperm is produced, spermatogenesis. This is taking place in the seminiferous tubulos. That is where sperm is produced. Based off of sperm production, the sperm will then eventually leave the seminiferous tubulo when it's mature and will make its way into kind of this vicinity right here, known as the straight tubulo, before eventually the sperm will go in the back of the testicle into an area known as the rete testes. And then from here, this is number 3, rete testes, the sperm will make its way in to an area known as the efferent ductule before eventually making its way into the epididymis. This area up here, this is the head of the epididymis. Eventually, sperm can go down to the body of the epididymis and eventually to the tail. And usually the tail is the portion that starts to really store the sperm. So looking at that again, the direction of sperm movement, sperm is produced within the testes in the seminiferous tubulos. Sperm then travels to the straight tubulo. Sperm then goes to the rete testes, to the efferent ductule, and then finally to the epididymis. And the epididymis, again, is the location where sperm is stored. This is the storage location for the sperm. Now, when the male ejaculates again during the sexual encounter, ejaculation, again, the epididymis undergoes very strong contractions and the sperm will then be expelled into the vas deferens. In this picture, the vas deferens is going to be right in the back here. So imagine then the sperm is going up through the tail, and here we have that vas deferens. And the sperm will continue on through the vas deferens, this tube-like structure, before eventually it makes its way to the ampulla bulb of the vas deferens. So here's a question here for you. What would happen if a male had his rhea testes cut, which is this area right here? If that was damaged, pretty much there's really no way the sperm can make its way from the testes over to the epididymis. And if that area gets damaged, very possible the man is no longer fertile. So I've known of men where that they've had an accident down there, either through baseball, very commonly if a baseball hits that area and damages that area, or even a hockey puck. That can definitely make the man no longer fertile, unfortunately. So, very interesting, I know. Sometimes some people are wondering, well, how long is the sperm viable? If the man's having sex, how long is the sperm viable in the female? If you've ever wondered about that, how many days is that? Approximately, it's about 5 days, give or take. A lot of scientific data shows the sperm being viable in the female for 5 days because of the environment in the female, the moisture there, things like that. It can't dry up. So approximately around 5 days or so. Some data is showing 3 to 5 days, but approximately around 5 days. So that's kind of how long approximately it can be viable, alive in the female. All right, this picture here showing histology. As you know, I like my histology. And we're looking within the testes in a structure known as the seminiferous tubules. And again, this is considered the site of sperm production, spermatogenesis. So each of these structures here, circular structures, represents a seminiferous tubule. So right here, where this arrow is pointing, right here, are seminiferous tubules. This is where sperm is produced, okay? Is in these structures, these seminiferous tubule. This is where sperm is produced. This is where sperm is produced. So near the membrane area here is where you'd have more of the stem cells, and then the steps of spermatogenesis occur. And you eventually get in the middle here, and this is where you'd have a mature sperm. So there's my little sperm there. I know I'm not the best at art, but right in the middle here, you'd have that mature sperm, just so you're aware. Okay? So seminiferous tubules, this is where spermatogenesis occurs. Another very important structure found in the testes is known as the interstitial cells of Leydig. The interstitial cells of Leydig. These cells is the precise location where the sex hormone for the male is produced, which is testosterone. Testosterone is produced in the interstitial cells of Leydig. So the interstitial cells of Leydig, they're going to be found outside of the seminiferous tubules. And in this picture here of histology, you'll see that right here. So here's a picture of the interstitial cells of Leydig. Again, this is where testosterone is produced is within these interstitial cells of Leydig. And this picture here, once again, is a seminiferous tubulo. Seminiferous tubulo where the sperm is produced. But right here, this is a picture of the interstitial cells of Leydig where testosterone is produced. So birth control for males. We always think of birth control that maybe women are taking, but this is definitely an option for men. And here we're looking at the procedure known as a vasectomy. So a lot of men do get this done. Maybe there gets to a point where mom and dad have decided they have enough children already and go ahead and have this procedure done. So the male will go in for the procedure where during the surgery you'll have the cutting and tying off of the vas deferens. So here's the before, vas deferens completely connected, sperm travels, and now here's the after where the vas deferens has been separated. This is fairly effective, close to 100%, but sometimes there can still be some fibers connected where it doesn't always necessarily work and you have to have the procedure again. But a lot of men have this done, and so that way that really prevents the probability of having another child. So a lot of men have this procedure performed. We now are going to start taking a look at the penis the next couple slides. And the penis is made up of several different parts. You have the root of the penis, the shaft of the penis, and the glans penis. So, root of penis, the root is that first initial part right here that protrudes off the main core of the man's body. And then the shaft of the penis right here, this makes up the bulk of the length of the penis, the shaft, this section. And then finally, the last part, the glans penis. And this is the most sensitive part of the penis because of a lot of the nerve receptors there. And this part's even more sensitive in a male that is uncircumcised. So all men, all boys I should say, when they're born, contain foreskin when they're born. The foreskin, the scientific term of that being the prebis. This is a loose skin that covers the glans penis, which is represented here in the picture. It would be right here and right here covering the glans penis. So the parents have to decide if they want to have their child circumcised. A good proportion of men in the United States are circumcised, where you remove the foreskin after birth. So that's a decision mom and dad have to make. There's definitely pros and cons to the circumcision. One of the big reasons why you'd have it removed is really just for easier personal hygiene to have it removed. And that's a big reason, along with decreasing the chance of STDs, infection, cancer of the penis, those types of things. Religious reasons too. But other parents decide not to because what if something goes wrong in the surgery? What if that area is destroyed? Something just goes wrong. And they feel like, the male's missing part of his body and, maybe he should make that decision later in life. There's studies that show that there's a lot more additional receptors during the sexual encounter to make it even more pleasurable. So, again, ultimately, the parents have to make that choice. Do we want to remove the foreskin or not? And again, it varies around the world, with some men being circumcised or not. Majority of men in Asia are not circumcised, along with many areas of Africa and even Europe. And even it varies in the United States with a lot of men out in the Pacific Northwest and California aren't circumcised. in comparison to men in the Midwest and the South. So there's a lot of variation even geographically in our own country. Okay, so continuing to look at the penis here. When we look at the penis, there are going to be several different erectile tissues of the penis. One type of erectile tissue here, number one, is known as the corpus sponginosum. Corpus sponginosum. And this corpus sponginosum surrounds the urethra of the male and also forms the glans penis. So on this model here, this image, it would be this area here where you find this erectile tissue, and likewise here in the glans penis for the corpus sponginosum. The next erectile tissue is made up of two erectile bodies, so corpora cavernosa, or just talking about one, corpus cavernosum. So the corpora cavernosa, two erectile bodies, makes up the bulk of the penis, especially the shaft of the penis. And that would be right here. So right here and right here is that corpora cavernosa. So of course urethra goes through the penis and also semen, but always at different times as we'll see going forward. And the penis, very critical during the sexual encounter where the penis becomes erect, the erection, where the penis is going to be erected because of the tissues that are engorged with blood. We're now going to take a look at the accessory sex glands of the male. So there are going to be 3 accessory sex glands that ultimately release their contents during the ejaculation, forming the semen. So we can kind of see these here on the diagram. The first structure here, number 1, this is known as the seminal vesicle. So seminal vesicle. So seminal vesicle, which is this like pea pod structure that you can kind of see here. I'll circle it here for you. We also then have the prostate gland. So here's the prostate gland. We'll likewise circle that here for you, so right here. And then finally, a very small accessory sex gland of the male is known as the bulbourethra gland. So that would be on the diagram right here. So you find that on the right and left side there, so that's why I have glands. Just like there's also 2 seminal vesicle glands, one on the right and left. There's just, of course, 1 prostate gland. Now we're going to take a look at and see what do these accessory glands expel during the ejaculation to form the contents of the semen. So first, starting at the top here, we have here what's known as the seminal vesicle, like we've mentioned before. So the items that are produced here, you have a sugar component for the sperm, the fructose. So this is some nutrients for the sperm. Also, believe it or not, vitamin C. So more nutrients for the sperm. A coagulating enzyme. Even prostaglandins. So even some lipids here. More nutrients for the sperm. The prostate gland, what it provides for the sperm is a nutrient component, which is called citrate, more additional enzymes, and what's known as the prostate-specific antigen, the PSA. So this is going to help to boost the immune system of the sperm. It also, the prostate gland components provide the milky overall composition of the sperm. And then finally, the bulbourethra gland. This will lubricate the glans penis and even neutralize traces of urine that could be slightly lower on the pH scale, possibly in the male urethra, and provides the thickness of the composition and a clear mucus. So altogether then, we form the semen. If you've never seen semen before, here it is in this Petri plate up here. So you can see a little bit of the milky complexion, the thickness of the composition. There's a little bit of a light yellow in there too. So again, some of the important things to consider when we look at the semen, what makes up it. Again, there's the nutrients, the fructose, the citrate, the vitamin C like I mentioned. There's also sometimes the hormone found within the semen known as relaxin, which can protect and activate the sperm. Another important idea to think about is the pH of the sperm, which is more alkaline, basic. You can see here that range of the pH, which neutralizes the acid that might be in the urine of the male, and especially the acidity of the vagina of the female, because the vagina has a very low pH, and so a lot of sperm actually die in the vagina. And then for the prostaglandins, these are the lipids I was referring to, and they can play a role with stimulating reverse peristalsis, which occurs in the female in the uterus. So this can facilitate sperm movement in the female. If you've ever wondered how much semen is actually ejaculated during sex, it's about 10% of the sperm of the male, so 2 to 5 milliliters on average. And within the semen, there's millions and millions of sperm expelled. Anywhere from 20 to 150 million sperm are released during sex. We're now going to take a look at the 2 autonomic nervous system control responses of the male. So the first one we're going to look at again is the erection. When the male experiences the sexual encounter, the penis will undergo the erection, where you have the engorgement of blood in the erectile tissues. So this erection is controlled by the parasympathetic nervous system. So the erection is controlled by the parasympathetic nervous system. And this picture kind of shows that. During this encounter, sexual encounter, there's going to be the release of nitric oxide, this overall chemical. And this causes the erectile tissues to actually fill with blood and have the erection. So when men get older, sometimes they might have a difficulty achieving the erection because of a decrease in the production of nitric oxide. Hence, there's different overall medicines out there on the market that some elderly men take so that they can have an erection. When we focus in on the erectile tissues, the corpora cavernosa is important for maintaining the engorgement of the blood in the penis. And the corpus sponginosum is important because it keeps the urethra open, which is critical for the movement of the semen. Eventually, the male will get to a point where the ejaculation occurs. And so the man will reach the climax during the sexual encounter. During the ejaculation, this is when the semen is going to be released from the penis. And ultimately, ejaculation is controlled by the sympathetic nervous system. The sympathetic nervous system. So ultimately, this is where we're forming the semen. Those accessory sex glands contract, empty the contents into the urethra, so forming the semen. And during this brief period of time, the bladder sphincter muscles constrict. So that way there's no urine release. It's just going to be semen going out of the penis. And the penis muscles also play a huge role in the release of the semen. They undergo very strong rhythmic contractions, and the semen releases during sex, and it's been clocked at a speedometer rate of around 11 miles per hour. So that's how fast the semen is being released from the penis during sex. And that fast rate's important to go into the female, the semen, quickly, to definitely increase the chances of progeny, of children. All right, we've reached our first set of review questions for today. So time to find your iClicker again. And our first question here today is, sperm is produced in the blank. So where is sperm produced? Sperm is produced in the blank. So we'll give you about a minute or so to go ahead and come up with the answer. All right, and the correct answer here would, based off these choices, I would probably go with choice letter A. So letter A would be the correct answer. Sperm is produced in the seminiferous tubules. Sperm is produced in the seminiferous tubules. Number 2 for review. Sperm is stored in the what? Sperm is stored in the what? All right, and the correct answer here would be choice. Here we are, choice letter C, the epididymis. Choice letter C, the epididymis. That is where the sperm is stored. Number 3 for review. During ejaculation, sperm travels from the ejaculatory duct to the blank. Where does the sperm travel next, based off of these choices? Okay, and the correct answer here would be choice letter C. The prostatic urethra is the correct answer here. Number 4, which of the following items are secreted from the seminal vesicle, forming semen? So what would be the correct answer here? And the correct answer here, I would probably go with choice letter D. Both A and B would be correct, right? Our vitamin C and fructose sugar. Would it be citrate, because citrate is produced in the prostate gland? All right, moving on to the second half of our notes today, we're now going to take a look at the physiology of the male reproductive system. So a good place to start is to have a good understanding of spermatogenesis. So spermatogenesis, a fancy term for producing the sperm. And again, the sperm produced in the seminiferous tubules in the testes. A healthy male will produce around 400 million sperm every day. And this starts right when he starts puberty, around the age of 14 approximately. So we break down the stages of spermatogenesis. And the first stage here is mitosis. And mitosis is referring to cell division, which occurs in all the cells of our body. And this was a cell division process that you would have learned in Anatomy 1 and 152. So it might ring a bell when you start thinking of those stages of mitosis, right? There's the interphase, there's the prophase, and so on and so forth. So this starts with the step of spermatogonia. Spermatogonia is just a fancy term referring to the actual stem cells, to the stage of the primary spermatocyte. From that point on, we go into a different cell division process, number 2 here, which is known as the process of the meiosis. So mitosis to the meiosis. And this is referring to the formation of gametes, sex cells. So this goes from the primary spermatocytes to the stage of the spermatid. And then the last part of this process of spermatogenesis is looking at what's known as the spermogenesis. So it's not spermatogenesis, but the spermogenesis, which goes from the spermatids to eventually formation of the adult sperm. So this is kind of an overview slide. We'll get into more in-depth structures and physiology coming up here. All right, so we mentioned this term already, the spermatogonia. And again, spermatogonia is referring to the stem cell. It's just a fancy term, spermatogonia. So that refers to a stem cell. So, stem cell. And spermatogonia is just a fancy term for that. So the spermatogonia, the stem cell, so you can see that up here, it's right along the border of the seminiferous tubulo here in the testes, will ultimately become either a type A daughter cell or a type B daughter cell. So the differences between the two, the type A daughter cell will go ahead and maintain the germ cell line. What that means is these type A daughter cells are not going to go through spermatogenesis to form sperm. They're going to be critical, so you always have stem cells, because if you have no stem cells, you ultimately can't produce sperm. So that's what they're going to do. The type B daughter cells are the ones that actually undergo spermatogenesis and will develop into the primary spermatocytes. That's what type B daughter cells will become, the primary spermatocytes. So that's what they do here, the type B daughter cells. This image here is taking a look at some very unique cells known as the Sertoli cells. They're also known as the sustenacular cells. And they're going to be the ones here, if I get the, here's my laser pointer. The sustenacular cells or Sertoli cells are actually the yellow you see here. And so they kind of assist with spermatogenesis, the different cell stages. Here's the nucleus of one here. So for their function, they're important for forming the blood-testis barrier to prevent antigens into the bloodstream that could activate the immune system. So that's important, definitely before puberty, because you don't want accidentally these cells to get released from this area into the male's body because they shouldn't be there yet, right? But they're important for providing nutrients, maybe some direction for the sperm production. They dispose of excess cytoplasm, which is important. And they help with transporting the sperm from one step to another, and ultimately just an overall assisting of spermatogenesis in general. This picture is wonderful. It breaks down every single step of spermatogenesis. So we'll go ahead and we'll go through this slowly here for you. So starting off, we start with our stem cell. And this is right up against that membrane layer of the seminiferous tubulo in the testes. So the spermatogonium, again, a fancy word for the stem cell. And from there, then mitosis will take place, just normal cell division, where you end up with two daughter cells formed at the end of mitosis. The type A daughter cell, which is critical for maintaining the germ cell line so that you always have stem cells in place. The other daughter cell formed is the type B daughter cell, and this is the one that's actually going to undergo spermatogenesis. So these early stages here, spermatogenesis, the process of development of making sperm, mitosis plays the critical role. Here, of course, we have the type B daughter cell. So from here, the type B daughter cell will be transformed into the primary spermatocyte. And at this point now, we enter into a new cell division step, now looking at what's known as the overall meiosis. There's two major divisions of meiosis. For mitosis, there's meiosis 1 and meiosis 2. During the meiosis 1, we start with the primary spermatocyte and end with development of the secondary spermatocyte here. So we go from the primary to the secondary spermatocyte and end up here with two daughter cells. Here are the two daughter cells. And you probably notice that there's a 2N versus an N. Maybe you noticed that, right? 2N versus N. What this is all about is really referring to chromosome sets, if you're curious. Okay, refers to chromosome sets. And that's what the 2N versus N is. So there's actually reduced a number of the chromosome sets from 2N to N. So that's ultimately critical to reduce the chromosome sets, because when you think of the amount of chromosomes in a new human being, you have half from mom, half from dad. So that's why that has to happen, because you get 23 chromosomes from dad and 23 chromosomes from mom. So hopefully that makes sense. All right, so we finished up the first division of meiosis, meiosis 1, and we're at the secondary spermatocyte stage. Now we're going to enter into meiosis 2. And in the second division stage, nuclear stage, we're going to start with our secondary spermatocytes, and ultimately they're transformed into early spermatids. So the secondary spermatocyte, and then ultimately that's going to be transformed into spermatids. So here's the early spermatids, and you have 4 daughter cells formed now. From here, the early spermatids will become late spermatids, and they almost resemble what we consider to be, sperm. Almost. They almost look like sperm. But we're not there yet. These late spermatids, they need to lose this excess cytoplasm. So eventually that will be shed, that will be lost. A lot of that can be picked up from those sustenacular cells that I mentioned. And then ultimately, we form our sperm. So here we have our sperm, our spermatozoa here. Okay? We have our sperm right there. So definitely important to know the steps involved with spermatogenesis. So definitely make sure to study this, because this might be important to know for your final exam. We're now going to take a look at the parts of the sperm. So there are 3 major regions of the sperm. There's the head of the sperm, which contains the DNA of dad in the nucleus. There's going to be the region called the midpiece of the sperm. This contains the mitochondria. That, of course, contains that really important energy component, ATP, which allows for lots of energy for the sperm to move and allowing for the tail to move around. And then finally, the last part here, this region's the tail of the sperm, which contains the cellular extension known as flagellum. Of course, this is important for propelling the sperm, almost like a propeller on a boat, so the sperm can easily swim from the male system into the female system. And then, of course, the head of the sperm contains a very unique structure known as the acrosome. And the acrosome is a helmet-like structure. So right here, a helmet-like structure of the sperm. And this contains hydrolytic enzymes. This enables the sperm to easily penetrate the egg and get through the outer layers of the egg for actual fertilization to take place. So this is the helmet-like structure. So the sperm has his helmet and he's ready for battle. So definitely important to know the different pieces of the sperm. So here we have a great picture of lots of sperm. And again, out of millions of sperm that might be released into the female during sex, many of these sperm are going to die. They're going to die in the vagina because of the low pH. They're going to be identified as a foreign item, an antigen, and many of them will die, potentially undergoing phagocytosis. Only a few thousand will survive. And then from there, many more will die. And maybe only 100 sperm will even make its way up to the uterine tubes, referring to the fallopian tubes, to actually fertilize the egg, the oocyte. And then, of course, out of those, maybe only one will be strong enough and fit enough to even make it to and through the layers of the egg to fertilize the egg. This graph here gives you an idea, again, of levels of testosterone in a man and likewise levels of sperm production. So approximately every day, a healthy male is producing 400 million sperm every single day. And with sperm production, we can see this on the graph here. It starts right when puberty occurs and then shoots right up. And this level of sperm production stays very, very high in the male, all the way up to 60, early 60s, mid 60s. So who's to say that, hey, maybe someone decides to become a dad later in life? Maybe. But sometimes sperm production can go down significantly earlier in the man's life, maybe around 40, and that's if the man's not staying healthy. If the male's involved with alcoholism, drugs, not eating healthy, not exercising, obese, sperm production may not stay as high in some man that's very healthy. So health of the male is very critical to keep the sperm levels high. Testosterone is indicated on the red line here. So you can see an increase in testosterone levels when gender has been recognized during pregnancy. And that's important to shoot up like that for production of the penis and production of the scrotum and testes and things like that. And then it can go down a little bit after that once those items are formed. And then it shoots up briefly right after birth. So that way there's a little further growth there and very key early on. And then it stays low, and then it really shoots up when it comes to a lot of the secondary sex characteristics of the male, the deep voice, hair on the chest, pubic hair, things like that. We're now going to take a look at the physiology involved with really ultimately the triggering of spermatogenesis. And a lot of this is ultimately starting up in the brain. When it comes to puberty, it all starts up in the brain, up in the hypothalamus of the brain, which would be up here, and then signals being sent down to the pituitary gland right here, to the anterior lobe, so that part there in the brain, and then signals being sent down to the testes. So we're going to go ahead and break this down on a step-by-step basis here next. So imagine that puberty has occurred in the young boy around 14, 15 years old. Ultimately, in the brain, here we have the hypothalamus. And the hypothalamus then is going to go ahead and release a hormone known as GnRH. So GnRH right here will travel to the anterior lobe of the pituitary gland in the brain. GnRH is an abbreviation for the gonadotropin releasing hormone. Gonadotropin releasing hormone. So this hormone now has made it to the pituitary gland, to the anterior lobe. And GnRH then will interact with the anterior lobe of the pituitary gland to go ahead and secrete two new hormones, FSH and LH. FSH refers to the follicle stimulating hormone, and LH refers to the luteinizing hormone. So those two hormones are secreted from the anterior lobe of the pituitary gland and will then leave the brain, travel through the bloodstream, down to the testes of the male. And here we can see that here, FSH and LH, they travel down to the testes. That's where they go. But they're going to go to different parts within the testes. So this is supposed to be referring to testes here. All right, where do these critical hormones go? The FSH and LH that we've been talking about. So of course they go down to the testes, but they go to different cells that they act upon. FSH will go ahead and interact with the sustenacular cells, which release the androgen-binding protein, ABP. It's going to make the spermogenic cells receptive to testosterone. So that's kind of what we're seeing in the image right here. Our other critical hormone, LH, the luteinizing hormone, interacts with the interstitial cells of Leydig, and that leads to testosterone being released here. So that really starts that triggering of testosterone, and testosterone ultimately is the final trigger for spermatogenesis. So testosterone is so critical in the male for maturation of his sex organs, his penis, his scrotum, the growth of those structures, overall maintenance development of secondary sex characteristics. So again, the deep voice, the hair on the male's body, his chest growth, things like that. We'll get into a little later, but very critical. But we ask ourselves, well, what would happen if the interstitial cells were removed? They're surgically removed. They never formed. Well, if that's the case, you might have a male that might have more of, maybe looking more female perhaps, more feminine characteristics. Because if those are removed, these cells, you'd have very low levels of testosterone here then. If per se those cells were removed or damaged, there's still some testosterone being produced because you have a little bit produced in your adrenal gland, but not a whole lot. Now, let's say that there's a situation where maybe there's significant levels of testosterone being produced. So we kind of look at proper overall regulation of hormones here. So if testosterone levels were increased significantly, this can lead to a release of a new hormone we haven't mentioned yet, known as inhibin. So inhibin is released when sperm count is significantly high, and the sustenacular cells we've mentioned before, they actually produce the hormone inhibin. So what happens then with this hormone inhibin being released from the sustenacular cells, per se here, inhibin goes through your blood. It leaves the testes, travels through the blood, and can actually go up to the brain. And temporarily, up in the pituitary gland, anterior lobe of the brain, it can actually inhibit FSH release temporarily, and likewise can inhibit GnRH from being released from the hypothalamus. So by doing that, this is going to temporarily stop testosterone production, get it down to a normal level of homeostasis. So here we look again at the important functions of testosterone, why it's so critical in the male. One, it's a big trigger for spermatogenesis to occur, for production of sperm. It also will target the overall growth of the accessory sex organs and glands, growth of the prostate gland, growth of the seminal vesicles, bulbourethra gland, even growth of the more of external genitalia, the penis, the scrotum. Testosterone's also going to play a role in the overall metabolism of the male. He's going to be eating a lot of food. Metabolism rate off the charts. And provides a nice basis for the sex drive in the male. Testosterone is the driver for sex drive in men. And here we take a look at the secondary sex characteristics which come about because of testosterone. So a lot of hair development, facial hair, armpit hair, pubic hair, growth of the chest, and deepening of the voice. Skin becomes thickened and oily. Overall bones will grow and increase in density and size along with skeletal muscles. And then eventually, you have your man, Conan the Barbarian, Arnold Schwarzenegger. There's your man. All right, so we end lecture today with a couple review questions. So time to find your eye clickers once again, and we'll go through these questions together. So, number 5, the blank contains ATP, which provides metabolic energy for whip-like movements of the tail. The blank contains ATP, which provides metabolic energy for whip-like movements of the tail. So what would be the correct answer here? What would be the correct answer here? And the correct answer here should be choice letter C, the midpiece of the tail, because that provides that overall metabolic energy because of ATP and found in the mitochondria. Number 6, a type B daughter cell will evolve into a blank during spermatogenesis. A type B daughter cell will evolve into a blank during spermatogenesis. So what would be the correct answer for this question? And the correct answer here should be choice letter D, the primary spermatocyte. So letter D is the correct answer here, the primary spermatocyte. Number 7 for review, the hormone blank stimulates the interstitial cells of Leydig to produce and release testosterone, which will promote spermatogenesis. So what would be the correct answer for this question here? What would be the correct answer here? All right, and the correct answer here would be choice letter C, the luteinizing hormone, LH. So very important to know your hormones for the final exam. there's a sheet in your course packet under lecture handouts that breaks down the functions of these critical hormones: FSH, GnRH, LH, inhibin. So definitely make sure to know the functions of these. And you'll see a lot of hormones when we eventually get into the female reproductive system in the future here. So, all right. Well, have a wonderful day, and I hope you enjoyed today's lecture.